is understanding the 100 trillion microorganisms that call us home. So the human microbiome has received a lot of attention over the last 5 years or so. Essentially, the microbiome is a collection of microorganisms that live on us and within us. And there's over 100 trillion members living on us, within our bodies, and the total weight averages about 3 pounds. So it's a lot of microorganisms that we're carrying around. Most of them are living within our guts. You can have up to 10,000 different species of bacteria, fungi, protozoa, viruses living within us at any given time. They actually outnumber us on a cell basis by 10 to 1, forming really complex, tiny ecosystems. So just looking at the palm of your hand, you have a very diverse collection of organisms living there. And they outnumber our genes by a factor of at least 100 to 1. They think that it even might be up to somewhere around 150 to 1. So if you think about it that way, we're actually only about 10% human. So it's kind of scary to think about. So you can also think of it as our human bodies and these microorganisms is a superorganism. And we actually need these organisms to function properly. So our skin and our mouths, you actually have more bacteria living within your mouth than there are human beings on this planet. So it's really crazy. And what they do is they kind of provide a barrier, like kind of a protective barrier, keeps harmful organisms from coming in, causing infections. In our gut, they're extremely important. Our microbes are really important in digestion. So the enzymes and proteins that we secrete alone will not digest the food that we consume. So these organisms have evolved with us in a very close-knit relationship in order to digest our food, help metabolize it, so they provide certain amino acids, nutrients, things that we could not get alone. They're very important in immunity. I'm going to talk about that a little bit later. And there's this very important brain-gut axis. So they find, like in germ-free mice that are essentially bred within the laboratory to not have a gut microbiome, their brains don't develop normally. The serotonin that's produced in your body is, a lot of it is produced in the gut. So they're even now correlating certain like depression, even autism. A lot of disorders are now being linked to the microbiota that we carry in our guts. So a big question is, how do you study your microbiome? How do you know what's living there, what they're doing? So we use these meta-omic analyses. So meta is just, Greek origin meaning with, across, after. And omics is the study of various collections of molecules, biological processes, and physiological functions. So these meta-omic analyses are just looking kind of just at the biology of everything that's there in a given sample, in your gut, in the skin. So what we do as scientists when we're studying the microbiome is take samples from different body sites and sequence what's there. The DNA, the RNA, to see who's there, what genes are they carrying, what are they expressing, how they respond to different factors, biotic factors, abiotic factors. You can look at what metabolites they're producing, what proteins they're producing. So we can actually do a whole lot. And it all starts with DNA. So I'm sure everyone knows what DNA is, but just a quick refresher. DNA is deoxyribonucleic acid. It's the hereditary material that we all carry in our cells. So you have 4 bases, adenine, thymine, guanine, and cytosine, and they form the double helix of a DNA molecule. And within a gene, each set of 3 nucleotides codes for an amino acid. Amino acids are strung together to create proteins. The human genome in any given cell has about 3 billion bases of DNA. It's quite a lot. Your average bacterial genome is about 2.5 million bases. And if you look, just a brief overview of how we go from DNA to proteins, you start with this double-stranded DNA molecule. In order to express a gene, RNA polymerase comes along, unwinds it, and what it does is it makes a single-stranded mRNA molecule. And from that mRNA, you code for proteins. And how that happens is these molecules called ribosomes. So they come along, read the messenger RNA, and kind of help synthesize proteins. And I go over that really quickly just because in order to understand what kind of microorganisms are living in any kind of sample that you take from the skin, like a skin swab, what we do is we do metagenomic ribosomal RNA gene profiling. So all we do is look at the 16S ribosomal gene. And this is specific for bacteria. So in humans, a similar gene is actually an 18S gene. Now, this 16S gene, when it's synthesized, is the major part of the ribosome. So it's extremely important because it's responsible for synthesizing proteins. So you don't want to have a mutation in that because it won't work properly. So, and because of how important it is, it's highly, highly conserved. So this gene sees very, very little mutation over time. And that's really good because we can use that gene to kind of identify certain bacterial species. And if you look at the 16S gene, it's about 1,500 base pairs. And what it has are these variable regions. And it can have 9 different regions within that gene that do vary from species to species. And what we do in my lab is look at V1 through V3, amplify it, sequence it, and then we compare that sequence to a database. And any sequence within that region that is 97% or higher, similar to another sequence that we find in the sample, that's the same species. So it's just a really quick, easy way to define a bacterial species as having 97% similarity within this region at the 16S gene. And once we have that sequence data, we can make these phylogenetic trees where you can look, from maybe a saliva sample, and anything that fits kind of close together on these little trees, well, they're really similar. They're highly related bacterial species. The further apart you are from something else, it's going to be something completely different. And there's, an unmeasurable amount of bacteria and microorganisms in the environment. So this just is a really kind of quick tool to understand the different bacteria that are living within a body site. Another tool we use is metagenomics. So this is whole genome sequencing. So instead of just looking at one gene in particular within an entire bacterial genome, what we do is we take a sample, skin sample, stool sample, and what you do, I'm just going to go over this really quick, is you actually cut up all the DNA within a sample, sequence it, reassemble it, and then blast it against a database to understand not only who's there, but what genes they carry. So it's a really cool tool. Other types of metaomics analysis that we use, metatranscriptomics, so something called RNA-seq. This is one that I'm actually heavily involved with at Jackson Laboratory, is that you can actually measure gene expression on a global level. So you can take the RNA, the messenger RNA, in a sample, and actually reverse transcribe it to cDNA, and then blast it against your databases. And you can also, so you know what genes are there and what level of expression. There's metametabolomics, so that's just looking at the small molecules that are present within a sample. And then metaproteomics, you can actually just look at the protein itself. Now, the Human Microbiome Project is something that my boss has been heavily involved with from the beginning. Started in 2007. And the NIH wanted to essentially have a whole bunch of scientists come together, work together, and characterize the human microbiome. So my boss is Dr. George Weinstock. He's actually been a leader in the genomics field from day one. He was involved in the Human Genome Project. He was one of the first to actually sequence a bacterial genome. And he has had a hand in sequencing everything from the honeybee to beetles to sea urchins to countless bacterial genomes. He pretty much knows it all when it comes to sequencing. So I feel very lucky to work for someone like him. So the Human Microbiome Project, what they did to characterize what kind of a normal, healthy microbiome is they got 300 participants, aged 18 to 40, and they were very strict about health, and they just took a whole lot of samples, over 12,000, from different body sites and sequenced them. And The advent of next generation sequencing has really moved the field along.